Hi guys, today we are going to present about Meselson and Stahl experiment. But before that, let's enjoy our nasheed.
رقت عيناي شوقا ولطيبة ذرفت عشقا فأتيت إلى حبيبي فاهدأ يا قلب ورفقا صلي على محمد.
السلام عليك يا رسول الله. السلام عليك يا حبيبي يا نبي الله. السلام عليك يا رسول الله. السلام عليك يا حبيبي يا نبي الله يا رسول الله. قلب بالحق تعلق وبغاره راء تألق يبكي يسأل خالقه فأتاه الوحي فأشرق اقرأ اقرأ يا محمد. السلام عليك يا رسول الله. السلام عليك يا حبيبي يا نبي الله. السلام عليك يا رسول الله. السلام عليك يا حبيبي يا نبي الله يا رسول الله.
يا طيبة جئتك صبا لرسول الله محبا بروضة سكنة روحي وجوار الهادي محمد.
يا طيبة جئتك صبا لرسول الله محبا بالروضة سكنت روحي وجوار الهادي محمد. السلام عليك يا رسول الله. السلام عليك يا حبيبي يا نبي الله.
Hey, did you know about the Meselson and Stahl experiment?
Yes, I know what Meselson and Stahl.
After Watson and Crick came up with their hypothesis regarding the structure of DNA, the scientific world was now curious to know how DNA could be replicated. They put forward a hypothesis that the DNA replicates in a semi-conservative manner, that is, both strands separate and each serves as a template for new strands. Templates, in literal term, means a pattern used as a sample to make something. However, the scientific world was not ready to accept this fact easily, so there were three theories put forward which hinted at the replication process of the DNA double helix. One was called the conservative model, the other was the dispersive model, and the last was the semi-conservative model.
Meselson and Stahl conducted the famous experiment in 1953 that supported the semi-conservative replication of DNA, a crucial concept in molecular biology. There is isotopic labeling to trace the replication process, confirming that each new DNA molecule contains one original strand and one newly synthesized strand. This experiment provided key evidence supporting Watson and Crick's model of DNA replication.
Wow! Wow! The first model in the list is the conservative model. This model suggested that two DNA strands separate and serve as template for replication. But soon after replication, the strands join back to form the original parent DNA, while the new synthesized strands join to form a completely new DNA. Thus, we get a newly synthesized DNA molecule, and we also get the original parent DNA molecule back, which is conserved. Hence, the name conservative replication.
Now comes to the next one. This was called as the dispersive model of replication. As the name suggests, dispersive means to disperse or scatter. So, what's get dispersed? Well, according to this model, the DNA which is about to replicate breaks into small fragments. The new strands get synthesized in between. As a result, we get a hybrid of both new and old strands together. The old parental strands appear to be dispersed in the newly synthesized one. Hence, this method is named as dispersive model.
And now comes to the last model. It was called a semi-conservative model of replication. This model suggested that two strands of DNA unwind and serve as templates for the newest strand. As a result, the replicated DNAs contain one old and one new strand, hence the name is semi-conservative. Nevertheless, this model was not accepted initially when put forward by Watson and Crick.
Now we are going to take a look about the methods of the experiment. In the experiments, bacterial cells which are E. coli were grown for generations in a heavy medium containing nitrogen-15. After that, these cells were transferred into a lighter medium that contains nitrogen-14 and grown for generations. Next, the bacteria were collected and the DNA was isolated. The DNA then was dissolved in a cesium chloride gradient. The tubes were then centrifuged at 100,000 G until equilibrium was reached.
After centrifugation, the E. coli cell samples will taken every 20 minutes, which is the generation time for E. coli cells growing at the optimal temperature. DNA with heavier nitrogen occupied the lower position in the gradient. DNA with lighter nitrogen occupied the top region of the gradient. This experiment involved 4 generations to determine which is the correct model of DNA replication. In generation 0, all of the DNA was in 100% heavy isotope because it has been grown in only N15 medium. After one generation in N40 medium, containing single band of DNA with 100% intermediate density, which is neither heavy nor light isotope. After 2 generations in N40 medium, 2 bands were obtained, which is 50% of the DNA was light density and 50% was in intermediate density. After 3rd generation in N40 medium, containing 70% DNA with light density and 25% DNA with heavy density. This showed that heavy strand was acting as a template for synthesized new strands with N40 isotope. Meselson and Stahl conclude that replications of DNA duplex involve building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
النبي صلى الله عليه وسلم ومن سلك طريقا يلتمس فيه علما سهل الله له به طريقا إلى الجنة